Now we have a set of early career talks. So these talks will be for 15 minutes each, followed by 5 minutes of question and answer sessions. The first talk is by Aniket Mitra from IIIT Bangalore. He will be talking about enhancing region-based geometric embedding for gene disease associations.
Okay, so I am a research scholar, MS student from IIIT Bangalore, and this is my topic, region-based ontology embedding for gene disease association prediction. Oh, okay. Thank you. Yeah, so these are the topics I am going to cover in brief. First, we will discuss introduction on what is ontologies and what is neurosymbolic AI. And then we will discuss a special embedding techniques called N-ball ontology embedding and use it for a specific biomedical use case of gene disease association prediction. Okay. So what is ontologies? Ontologies are machine readable form of vocabulary which can represent relational information. Okay. So owl ontology or web ontology language, which is expressed in RDF or XML format, is a very popular way to represent this kind of biomedical information. Okay. As you can see down, that thing is an owl axiom, okay, owl class. Okay, it can be converted into a description logic axiom. Okay, that's the logical form of that particular statement. So you can apply logical algorithms on it to deduce new information. And on the right hand side, there is the natural language representation of what it means. Okay. So what are the advantages of ontology? First of all, it is machine readable, obviously, so you can process it using some software or programming language. Secondly, it preserves the relationship between the related components in the ontology. Okay. Third, it is data consistent, that is, there is no erroneous data in the ontologies. Fourth of all, each and every concept in the ontologies are unambiguous. So everything is unique, there is no ambiguity in the data set. So as you have seen, it can be represented in description logic form. So you can use traditional symbolic logic algorithms to perform reasoning, that is deriving new facts. Also they are interoperable, that is we can use it seamlessly across multiple systems without any issues. Okay. So because of all these advantages we have seen, it is popularly used in many critical use cases, and one of them is obviously biomedicine. So what you can see on the screens are the various ontology repositories where different kinds of ontologies are available. In our lab, we mainly work with the foundation ontologies, that is the obofoundry ontology, which you can see in red. And the most popular ones among them are the gene ontologies, which contains all the genetic informations, and the human phenotype ontology, which stores the information about the different phenotypic abnormality observed during a disease. Now, if we just express the ontological information into description logic and perform traditional symbolic reasoning, the problem with that is that sometimes these ontologies are semi-perfect. Okay, so they are curated from different web sources, or there can be human error when they are curated by a human being. So this traditional algorithm fail on semi-perfect data, it needs to be perfect. Secondly, the traditional algorithms take a lot of time to converge, that is to perform the reasoning. That's why application of neural networks into the models, application of neural networks can enhance the performance, make it more robust. But the challenge is that we need to first embed this ontological information into vector space, that is convert them into numbers. That's what the neural network understands. So that's the challenge. Why? Because of the three reasons we have given down. So first of all, ontologies contains relational information. So the relation should be preserved in low dimensional vector space. Secondly, it contains logical information, that is subsumption there exists, which again need to be preserved. Thirdly, the ontology contains many topological information like the tree hierarchy or DAGs. Again, we need to preserve all this information for correct reasoning. So we have come out with the N-ball ontology embedding method and used it for the use case of gene disease association prediction. Okay. The data sets we have used is the human phenotype ontology. The link for the ontologies is given down where you can download it, it's publicly available. Apart from that, we have used the human phenotype annotation, which links the gene and disease information to a particular term in the ontology. And we have collected some gene disease association information from the publicly available database DisGeneT. Okay, let's first discuss a brief about what this n-ball algorithm is. So basically, in simple words, each and every concept or each and every phenotype from the ontology can be represented as n-dimensional ball in vector space. So you can see the C can be represented if as a 3D ball, then it will have a radius of size 3, it will have a center of size 3, that is 2, 4, 7 in figure 3, and a radius of 3, okay. So every concept can be represented as an n-dimensional ball in vector space, and roles can be represented as n-dimensional translation vectors. So basically it's a distance-based embedding model. So related informations, for example, a gene and a disease which are related, they should be close to each other in vector space. Okay. Now for example, let's take the axiom in figure 1. It's a simple axiom, B is a subclass of A. This can be represented in this form that the ball of B should be engulfed completely within the ball of A, as you can see in the figure 1. Okay. Similarly, another axiom if we take, okay, it includes a relation R. So what it means is that when C is translated by R, the new position C dash should be as close to D as possible. Okay. So this is the main working principle of the n-ball algorithm. Okay. Now we have designed some mathematical functions which preserves the meaning of these logical axioms, so that when we convert them into low dimensional vector space, the meaning of the axioms remains intact. There are many mathematical functions that has been discovered. There are 3 of them which we have shown here. Okay. The first one is the simple one, the B is a subclass of A. So from the equation, it is quite evident that what we want is that they should be close to each other, right? So the first part of the equation in figure 4 ensures that the ball B and ball A centers are close to each other, and the second part ensures that the radius of the ball A is greater than the radius of ball B, so that ball B is completely engulfed by A. The second one is that when C is translated by radius R, okay, sorry, by role R, then the new position C dash should be as close to D as possible, as you can see here, and the loss functions ensures that. Let's discuss another one in figure 6. So roles can be also subsumed. For example, let's take a very simple example. For example, if somebody is a parent, somebody is a father, obviously is the parent of that person. Okay. So this is called role subsumption. Now roles, what they are doing, they are translating the concepts from one place to another so that the related balls come together, right? So obviously both of them should translate in such a manner. In case of role subsumption, both the sub-role and the super role should translate the concepts in such a manner that their positions are not changed. Okay, that's why they should be close to each other, and obviously they should translate in the same direction. Okay, that's why 2 parts are there in equation in figure 6. First part makes the relations close to each other. The second part, 1 minus cos theta, makes them point in the same direction. So this is the overall architecture diagram what we have designed. So basically it's very simple. First we take the ontology in OWL format, convert it into description logic axioms. Then the description logic axioms we take the concepts and the relations and randomly initialize them in vector space. Okay. Then we train a neural network model based on all those optimization functions which we discussed in the previous slides. There are some more, and the embeddings reaches the final value. If our embedding technique is good and if it is properly trained, then ideally the related concepts should come close to each other. For example, a gene is related to a disease, they should come close to each other in vector space. So this is the overall working idea of our n-ball algorithm model. So we have tested our embedding model with other knowledge graph embedding models to check its performance. The evaluation metrics which I have used are the hit score and the k percentile rank. So it's very simple. Suppose we are doing gene disease prediction. So for a particular gene, we are ranking all the diseases based on the Euclidean distance between them in vector space. Since we know that ideally our correct gene and disease pair should have the lowest distance between them, so if we rank them in ascending order, obviously the highest one, the first one, the original disease which is linked to the gene should come at the first place, its rank should be 1, right. So this is the principle of the Hits K and the K percentile rank evaluation matrix. So this is the overall evaluation table. So it shows that our model has performed quite well in performing the gene disease link compared to other state-of-the-art embedding models. So finally, what we have also tried to do is that we also wanted to know if our model is quite good at distinguishing between the positive links and the negative gene disease links, that is incorrect links. And you can see that we have performed manually used statistical test, and there is a clear difference in the distribution of the positive and the negative links, showing that our model is quite good even at detecting the spurious or the incorrect links too. So we can conclude that our model is very well in embedding the complicated ontology information into vector space by preserving its semantic structure and the knowledge information. These are some of the work from our lab, and thank you.
Thank you for the talk. So one question, if you were to sort of use these existing language models which exist and use their embeddings, does it differ a lot from the embeddings that you get from your neural network, because it's fine-tuned on, or it's in a way you've designed the loss function to capture those 3 metrics which the LLMs would not? So I'm just curious.
Yes, yeah, so that's the main thing. So in our case, the loss function is particularly designed to capture the logical information and the topological structure of the knowledge graph. So if a particular LLM which is designed on a lot of data, if it's not designed on that kind of data, it generally fails to perform the reasoning on this kind of particular use case. So that's the thing. And current work of our lab is also evaluating the LLM models, so how they're performing in bioontology reasoning.
Thanks.
So ontologies, as you know, they are ever developing and are mutable. So did you also try to check the mutability of the ontology graph and see how robust your method is?
Yes, so yes, the ontologies are mutable, I mean they are changing, but currently it is static, okay, we are not, we are downloading at a particular point of time, then we are doing. So currently it doesn't take into account the new changes ontologies we are adding to the ontologies, so yeah, it's static as of now, the algorithm.
Did you try to do some test by inducing such changes to see how robust it still remains? Are some tests also done on those fronts?
Currently no.
Okay.
Yeah.
I'm just curious about the case of when a group of associated genes work together. For example, there could be a cluster of genes which is involved in this ontology. So in that cases, there might be many clusters.
Yeah.
Within a mega cluster we can consider. So are those things also included? Any sort of, for example, there could be 100 genes which are totally differentially expressed, right? And then there could be 10 clusters. Some of them would be in one pathway, some of them would be in different pathways. So how does that overall network looks like in this vector space?
So currently it is, the model is developed in such a way that it only includes the logical, it only tries to interpret the logical axioms in low dimensional space, but all these things you are telling, that genes clustered together and all these complexities, this is particularly, as of now, is not looked upon while developing the model. So yeah, these are some of the future challenges. If we can improve, we could get even better results.
Because you could kind of potentially use it to make a map of the whole disease, that could be a potential application. If with ever changing details we are getting from multi-omics data and all, this could be the things you could be adding.
Yes, yeah. Currently, we haven't done anything extra for the clustering part, but yeah, it's a great idea.
Hello, very nice talk. We have gene ontology databases. So, how like your databases is different from your designed LLMs for ontologies?
Yeah, so basically the gene ontology is available in many formats.
Yeah.
We specifically use the OWL format, Web Ontology Language format. It's a RDF kind of format, okay, see, this kind of format it is, okay.
Okay.
It's XML based. We try to convert it into a description logic axiom in logical phase, and then create and then use our model to do the reasoning.
So the model will just we need to provide our unidentified data, and then from those data from your LLM, it will process and it will provide us the ontology of those desired thing. Is it so?
Yeah, so basically you will provide the ontologies in logical form, and yes, it will perform the reasoning and it will bring the related information closer in the vector space. So yeah, if you are now trying to do a new query that whether A is related to B or not, you can use our model and it will give you some answers.
Okay, thank you.